Awesome. Hi, my name is Hannah Bradley. I'm a PhD student at Oakland University, and today I'll be talking about antiferromagnetic artificial neuron modeling of biological neural networks. So first, why are we using antiferromagnets? Well, because of their anti-parallel spin patterning, AFM materials have a natural frequency in the terahertz range. This means we can use them to create very fast devices. These frequencies can be excited by a spin current applying torque to the magnetic sublattices of the AFM. The exchange energy between the sublattices changes, which allows for these terahertz frequencies. The spin current acts on both magnetic sublattices, causing them to rotate. But by using biaxial anisotropy AFM, the rotation is not uniform in time. The rotating spins create a spin pumping signal, the magnitude of which is proportional to the rotational speed. And non-uniform rotation at a terahertz frequency results in a sequence of very intense, very short spin pumping spikes. The orientation of these sublattices can be described by using the angle phi, which is the angle between the Neal vector and the EZ axis. The derived equation for phi resembles an equation of a mechanical pendulum under a driving force, with the driving force being the spin torque from the spin current. But the most important part of this equation is that the exchange energy contributes an effective mass. This means the oscillator now has inertia. The oscillator can now act as an inertial particle under a driving force, with the driving force being the spin current. When the current is at a supercritical level, the AFM will act as an auto oscillator, continuously outputting spikes. But let's consider the case where the current is at a subcritical level and not enough to send it into an oscillation alone. With an additional pulse of current, the oscillator can act as a neuron, overcoming the energy barrier and causing a single oscillation. At the bottom of this sinusoidal path, the change in the angle phi is the fastest, which results in an output spike due to spin pumping. Like all systems with inertia, the rate at which this particle goes through an oscillation is dependent on the strength of the driving force and the additional stimulus. Now, the oscillator can act as a very fast artificial neuron, always ready to go and to be set off by an external signal. And they are unlike conventional artificial neurons because of this inertia. We need to be able to connect AFM neurons together such that they can be set off by one another. In our simulations, we just use an arbitrary coupling matrix to determine these connections. Linking up multiple AFM neurons in a chain allows the output of one neuron to act as an input for the next. So an output spike can travel like a wave through the chain. In conventional artificial neurons, there's an immediate reaction as a result of an input. But in AFM neurons, there's a delay between a single neuron receiving input and the resulting output. So how can we harness this delay in any meaningful way? Well, it'd be nice if we could create a system like biological neural networks where an output can be suppressed by a second input. So with a single input, there would be an output, but with a second inhibitory signal, the output would be suppressed. Such a circuit would not be possible with conventional inertialess artificial neurons. But it is possible with AFM neurons. We can create this inhibitor circuit by splitting the input into two and controlling the coupling of the output neuron. So the output neuron would need to receive a signal from both blue intermediate neurons in order to fire. In the first case, we see a single input causes the two blue neurons to fire at relatively the same time. This means the output neuron receives a strong enough kick needed to fire. But in the second case, the red inhibitory spike causes one of the blue neurons to fire early. This means the output neuron does not receive the two signals at the same time and does not have a strong enough kick needed to fire. Such inhibitory behavior would not be possible with inertialess neuromorphic circuits. So now let's use AFM neurons for a unique purpose. The biological neurons in our body make up thousands of different neural networks, all uniquely designed for different tasks. And we know AFM neurons have features that resemble biology, like inhibition, but also other things like refraction time and bursting. So let's see if we can simulate a biological neural network with AFM neurons. Creating biologically realistic neural networks with artificial neurons have applications in the future of bio-inspired artificial intelligence. For example, AFM neurons run on a much faster timescale than biological neurons, picoseconds to milliseconds. This means when we create these biological neural networks with artificial neurons, the robots we implement them in will run much faster. So in the future, when I build Terminator, he's going to have much faster reflexes than you do. The neural network we would like to simulate is the neural network responsible for the withdrawal reflex. This reflex is what happens when you might touch something hot and involuntarily move your hand away. The withdrawal reflex works by simultaneously contracting agonist muscles and inhibiting the extension of antagonist muscles in the same limb. Simply put, it just contracts one muscle and relaxes the opposing muscle to move a limb away from a harmful stimulus. The biological neural network, called the reflex arc, is made up of 4 parts: the sensory neuron, the brain, interneurons in the spinal cord, and motor neurons. And there are 4 scenarios that define the withdrawal reflex. The first scenario is simply voluntary motion. This is when we just consciously make the decision to move our arm. Next is the reaction to a weak stimulus. The strength of the stimulus is encoded in the frequency of the sensory signal, similar to biological neurons. With a weak stimulus, meaning something not very painful, the signal will travel through the brain to the motor neuron. However, if the stimulus is strong, there we go. However, if the stimulus is strong, the information will travel through the interneurons to the motor neuron. And finally, our inhibition of reflex is when the sensory signal is strong enough to trigger the reflex, but we consciously make a decision to counteract it. The AFM neural network will simulate all 4 parts of the biological neural network: the sensory neuron, the brain, interneurons in the spinal cord, and the motor neuron. There are 2 interneurons that will either excite contraction in muscles or send inhibitory signals causing muscle relaxation. And there are 2 motor neurons that correspond to muscles that either need to contract or relax. Here's a more detailed picture of the neural network, and we can see that the inhibitor circuit is used multiple times. For the interneurons, the weakened coupling of the inhibitor circuit ensures only strong sensory signals with a high frequency can excite the interneurons and trigger the reflex. It also allows signals from the brain to inhibit signals for the interneurons for the reflex inhibition scenario. The motor neurons are also inhibitor circuits, and this allows signals from both the brain and the interneurons to send exciting signals for muscle contraction, but also inhibition signals for muscle relaxation. Even with no sensory stimulus, our brain is constantly sending signals to the muscles to keep them engaged. This is called the tone. To make our simulations more realistic, we simulate this tone by having the brain constantly send a low-frequency signal to the muscles. This signal is created with an AFM neuron with a driving current above threshold, such that it acts as an auto-oscillator, continuously outputting spikes at a low frequency. The first scenario we simulate is voluntary motion. Voluntary motion starts with a signal from the brain with no sensory input. Again, this signal is created with an AFM neuron acting as an auto-oscillator, continuously outputting spikes. The signal from the brain travels to the motor neuron, causing the agonist muscles to contract and the antagonist muscles to relax. And we see this in the spiking pattern of the motor neuron. A spike indicates a muscle was excited and therefore contracted, while no spike means relaxation. Next is the reaction to a weak stimulus. Now the sensory neuron is an auto-oscillator, outputting spikes at a relatively low frequency. The sensory neuron sends its signal to the interneurons. However, because of the low frequency, it is not strong enough to excite the interneurons and trigger the reflex. Instead, the sensory signal must travel all the way through the brain to the motor neuron. This causes a large delay between the neural network receiving stimulation and the resulting reaction in the muscles. The reaction to a strong stimulus starts in the same way as the reaction to a weak stimulus, with a sensory neuron as an auto oscillator. However, now it is outputting spikes at a much higher frequency. The sensory information is now strong enough to excite the interneurons and trigger the involuntary withdrawal reflex. The sensory information bypasses the brain, travels through the faster interneurons to the motor neurons. Now the muscles react over 2 times faster compared to the reaction to a weak stimulus. This is the withdrawal reflex in action. The biological withdrawal reflex in our bodies takes about a half a second to move a limb away from a harmful stimulus. With AFM neurons, that time scale is on order of nanoseconds, highlighting the advantages of AFM neuron modeling. The last scenario is inhibition of a reflex. For inhibition of a reflex, we start with the same strong stimulus as before, which is strong enough to excite the interneuron. However, now a signal from the brain interrupts the signal from the interneuron. To fully counteract the reflex, the signal from the brain must cause the opposite reaction in the muscles. The agonist muscles relax and the antagonist muscles contract. And we see this in the spiking pattern of the motor neuron. So as a summary, I've shown that AFM materials can be used to create ultra-fast neurons with an effective inertia. And that effective inertia can be used to create biologically realistic inhibition through the inhibitor circuit. And then finally, we can use AFM neurons to simulate biologically realistic neural networks, such as the withdrawal reflex. In particular, the ability to simulate inhibition and inhibitory interneurons is a unique feature of AFM neurons that would not be possible with conventional artificial neurons. The last thing I'd like to mention before I go is that in June 2024, Oakland University will be hosting the International Conference for Microwave Magnetics. There's also an ad in Gathertown that you can look at for more detail. Abstracts open December 1st, and I hope to see all you guys there. Thank you for your attention.
Thank you, Hannah. Very interesting talk. So is there any question? Do you know any specific materials that can be proved as antiferromagnetic with this?
Yeah, our simulations use nickel oxide on top of platinum. They're all simulations that's theoretical at this point, just because creating AFM thin films is difficult. But yeah, we use nickel oxide parameters.
Okay. So, any other question?
I had a question real quick.
Go ahead.
What is, say, like a follow-up to this? Are you going to continue modeling biological circuits? If so, what do you have in mind?
Yeah, so this project was started with a medical student coming to our group who had a background in neurons. And we picked this withdrawal reflex to model just because it was the simplest one to start with. After the withdrawal reflex success, which I presented here today, we are thinking about moving to other biological systems, specifically cardiac neurons, so how neurons in the heart work. So hopefully that will be our next project.